Yeah, okay, so the main idea of this project is that, you know, because the surface of the ice sheet is getting darker, and we know that it will probably cause more melting, which obviously causes problems, so people are interested in this. And then in the last couple of years that we found out that actually this darkening of the surface might actually be helped by biological processes, by microbes growing and producing dark substances, and so on. And there's like a circle that this project is aiming to look at, and that's because there's more melting, there's more liquid water, and the duration of the melting season is longer. So there's more sort of space or more time actually for the microbes to grow and to produce stuff. So that we expect more and more microbes to be growing and to be present on the surface of the ice sheet, and then it can sort of cause or help darkening process, which in turn will sort of feed back into this loop. So that's the main idea of this project. So what we want to do within this project is actually we want to go to different geographically sort of different sites on the Greenland ice sheet. And also, it would be great if we could go to sites with a different albedo and different albedo change in the last years. And then we want to simply collect samples of the surface ice and the surface debris, craconite, and surface snow, and then simply to look at the numbers of the microbes in there and the diversity of the community there, and then use all the physical data available that the glaciologists can provide us from the weather stations, for example, from the ProMice project, which actually is the main collaboration for this project, and to do some sort of multivariate analysis, whether we can discern the main variables responsible for the changes in the microbial abundance and diversity.
Do you have any idea how important the change in microbial activity could be to albedo? Is it 1%? Is it 5%?
At the moment, we have no idea at all. We just know we know the concept that it happens this way, but we have no idea about numbers. That's the thing. That's one of the main reasons why we want to do this, because we can't quantify these processes yet.
What kind of work do you expect to do in the field?
Well, for us, it's mostly collecting samples, and then we need to freeze them and transport them to the lab, and then the real work is done in the lab when we look down the microscope and we can count, enumerate the cells, and we can extract the DNA and look at the DNA and the diversity of the community in the various microenvironments from the surface of the ice sheet.
What do you know about this biological this biome? What?
Well, we know a lot actually, and we know that it's the microbial community there is quite rich. It's quite diverse, and it consists of microbes that can fix CO2 from the atmosphere and build or convert it into organic matter. And there's others that actually feed on this organic matter, so sort of closing the circle. And there's lots of allochthonous organic carbon coming or being deposited on the surface from different sources, be it adjacent tundra, or be it sort of long distance organic carbon you get in aerosols. And then we know that the processes that the microbial growth actually can contribute to aggregating of the debris on the surface, because the microbes produce sticky substances which sort of glue the particles together, and they help form craconite holes, and that obviously has some effect on albedo. And they also, because you can imagine that it's a very bright environment. And so there's lots of radiation which can be harmful for the microbes, because it sort of it damages DNA, for example. So these microbes need to protect themselves from excess radiation. So some of them produce dark substances, as screening pigments. And that's the case of the now famous or infamous ice algae, which produce this really dark red pigment, and they can reach quite high abundances on the ice surface. And we think this can be actually one of the sort of major processes in darkening the edges of the ice sheet.